everyone. I'm Sarah Flanagan, and I'm here to talk to you today about the evolution of pigmentation genes in fish. But I'm actually talking about the evolution of pigmentation genes in pipefish, so hopefully that doesn't change anyone's opinion about coming to this talk today. Before I jump into it, I wanted to acknowledge how collaborative this work is. It wouldn't be possible without data and analyses done by my collaborators. And yeah, they've done all the actual sequencing and stuff, so so we all know that color is important. It's important in camouflage and defense against predation, in communication with conspecifics and heterospecifics, and of course in communicating with potential mates and trying to find mates and all of that. And we also know that these color patterns are the result of pigmentation cells and their ongoing interactions. There are two major types of pigmentation cells, the light reflective ones, such as iridophores and leucophores, and light absorbent cells. And fish are a great group to try to study these pigmentation cells because they have up to 6 different types. The 3 most common ones are the ones that are on the top of this slide, the iridophores, melanophores, and xanthophores. There are 4 main types of processes that these cells use to end up with the color patterns that we observe. So there's cell migration and differentiation, and the proliferation and interactions between cells to create the patterns that we see, plus of course production of pigments. And so given the important role of color patterns in ecological behavioral contexts, we might expect that genes involved in color patterning might be experiencing positive selection, particularly in species experiencing selection on those color patterns. So I'm focusing for this talk on the Gulf pipefish, which is a marine fish with sex role reversal, where female stripes or bands are under strong sexual selection. So here I'm showing you two females, and they have these blue iridescent bands that run dorsoventrally along their bodies. And we know that these are under strong sexual selection. We also might guess that the blue iridescent patterns might be the result of iridophores and maybe leucophores, which typically produce this type of color pattern, and that the background color might be a result more of xanthophores or leucophores. Another reason that I'm focusing on the Gulf pipefish is that there's a pretty decent published genome for them, which really facilitates genomic comparisons. So to ask my initial question of do pigmentation genes show evidence of positive selection in pipefish, I downloaded gene sequences for about 40 pigmentation genes from 10 other fish species. I used those sequences to annotate the pipefish genome for those genes, aligned those gene sequences, and used the branch site model to test for positive selection in the pipefish. Now, I'm going to warn you that these data are very preliminary. They came off of my computer last week, so there's much more work to do to finalize these results. So the first thing that you might notice in this figure is that I have 3 categories of genes, ones that were not annotated in my pipefish genomes, ones that were not significant, and ones that were significant. So I had a lot of trouble getting decent annotations and alignments in my initial pass with these genes. There's a lot more to figure out. But with the genes that were able to be analyzed, I found that the ones with significant signatures of positive selection were in iridophores and melanophores, not so much in the leucophore and xanthophore pathways. If I think about it instead in terms of those different cellular processes, I observed that there were some significant results in the differentiation and pigmentation, but you might also notice that I didn't really annotate any in the migration and proliferation categories. So as I said, very preliminary and a lot more work to be done. The other thing about this is that these gene sequences may or may not actually be involved in producing those pigmentation patterns in the skin. These are sequences from the genome. So I wanted to find out whether my pigmentation genes have skin-biased gene expression, and whether those putative pigmentation genes are actually being expressed in the skin of these fish. And I was able to answer this because my collaborators have this excellent set of transcriptomics data sets where we have data from 5 males and 5 females for 4 different tissue types: the gonads, liver, brain, and skin. And these transcriptomes are for adults. And so we might not expect that all of the pigmentation genes would be expressed, because some pigmentation genes are more likely to occur during development of embryos and becoming adults as opposed to expressed as an adult. However, the proliferation and pattern forming, the communication between pigmentation cells and the production of pigments is expected to occur as adults and occur as and be producing the signal that's actually under sexual selection, those band patterns. So the first result of this is that I found that pigmentation genes, the small set that I was able to recover, are enriched in skin relative to the brain. So what this figure is showing is along this axis here is a differential expression score, and on this side are genes that are more expressed in the brain relative to the skin, and on this side are genes that are more expressed in the skin relative to the brain. And each one of these bars is one of the many transcripts in my transcriptome. On the top here is an enrichment score, and the dotted line is sort of the null expectation, and the solid line is the enrichment score for the pigmentation genes. And the test for this analysis is significant. They are enriched relative to the brain. However, in comparing the other data sets, that's not a significant enrichment. And part of this may be because only 17 of the pigmentation genes that I was looking at were actually found in the transcriptome. So it's a pretty small, small data set to be testing things. And I plan to expand my analysis to a larger data set in the future. So I'm going to show you a heat map in just a second, but I want to kind of orient you to what you're looking at here. On this x-axis are the individual samples grouped by tissue type. They're also not particularly grouped by sex, so I have this color bar up here to show you which ones are males and which ones are females. The lighter colors are the females, the darker colors are males. I've added arrows to try to help, but I'm not sure if it does. And then on the y-axis, I have the pigmentation genes, and those are grouped by expression, clustered by expression profiles. So here's what the heat map looks like. There aren't really super strong patterns, but I'll highlight the skin ones for you. And so one of the questions I wanted to know was which processes are these pigmentation genes actually expressed, which processes are these genes associated with? Am I actually seeing that expected pattern that proliferation and pigmentation genes might be more likely to be expressed in these adult skin tissues? So there were 2 of these genes were involved in proliferation, or are known to be involved in proliferation, but note that there are multiple isoforms for these 2 genes. And I also found that 10 of these genes are involved in pigmentation production. Again, multiple isoforms, and so I think there could be something really interesting going on with the fact that these genes seem to have alternative splicing, or that there are some sort of gene duplicates that I'm not able to pick up on with my analysis at the moment. So this is something I'm going to be looking into. One of these proliferation genes showed a signature of positive selection from that analysis of the comparative genomics, and one of the genes involved in pigmentation showed a signature of positive selection. Another one of these genes is associated with differences between the different pigmentation types, but most of the genes that I found were experiencing positive selection are not actually found in this, were not actually annotated in transcriptome. So given this, I thought maybe it would be informative to look at the transcriptome sort of more broadly, without focusing just on the pigmentation genes. And I found that the skin transcriptome is most, so here is a multivariate space with the transcriptome samples on here. The skin ones are in red with the red upside down triangles, and the filled in points are the males and the empty points are females. And there are two patterns that pop out to me. One is that the skin transcriptome is most similar to the gonads in this multivariate space, and also that the males and females seem to be broadly overlapping. So I was able to investigate the KEGG pathways that are enriched in the skin, and there were a number of them that popped out. Don't worry too much about what all of them are. Here on the x-axis is a expression score, so if it's to this right side, the genes are upregulated in the skin relative to the other tissue. To the left, they're downregulated. And for each one of these categories, there will be 3 different comparisons: the skin versus the brain, gonad, and liver. And the first thing that pops out when you look at this really overwhelming diagram is that there are generally, most of these pathways are not shared across all 3 of those comparisons. For instance, melanogenesis, a pathway I would have expected to be really important for looking at pigmentation genes, because melanogenesis is involved in the production of melanin. It's only enriched in the skin relative to the brain. The 2 pathways that are enriched in all 3 are focal adhesion, which is not all that surprising because focal adhesion is involved in anchoring skin cells to the basement membrane. So in this diagram, these little proteins here are holding the epithelial cells to the basement membrane. So this is sort of a sanity check. Hey, skin-related genes are enriched in the skin. That's kind of cool. And then the other interesting pattern that popped out was that the herpes simplex infection pathway is enriched in the skin, which to me implies that the skin is an important part of immune defense in pipefish. So to summarize, the skin transcriptome reveals potential evidence for selection, potentially sexual selection, by showing some amount of positive selection on iridophore and melanophore genes. The pigmentation genes may be enriched in the skin, but the skin transcriptome is revealing other pathways that are important in the tissue. Before I finish up and take questions, I want to say I'm hiring. I'm looking for a PhD student to come work with me on projects like this. So if you are interested, or if you know anyone, please send them my way. So with that, I'll take questions.
So for your skin transcriptome, is that when they're not in like the breeding coloration, so the females, when they're like standard colors?
Yeah, so the question was whether there's breeding coloration that would be transient, and so it's more. So Drew, my collaborator Drew is the one who collected it, and I'm fairly certain that it's sampling the not transient color patterns, but the more